Chapter 3: Cells and Tissues. Let's talk about energy in biological systems because, let's face it, every living thing needs energy to survive. For plants, they're like nature's solar panels. They capture radiant energy from the sun and stash it away in chemical bonds, like a savings account of energy. For animals, well, we don't have that luxury. Instead, we get our energy the old-fashioned way, by eating. Whether it's plants or other animals, we import energy to keep our systems running. Let's dive into how energy flows through the environment. It's all about balance and transfer. The sun is the ultimate energy source, providing radiant energy that plants capture through photosynthesis. During this process, plants convert sunlight into chemical energy stored in biomolecules, like glucose. When animals eat plants, they tap into that stored energy. Their cells perform respiration, breaking down those biomolecules to release energy for work or store it for later. But there's always some energy lost as heat. It's a natural part of the process. The cycle continues as plants and animals interact with the environment, maintaining a balance of inputs and outputs, like oxygen, carbon dioxide, and water. Let's talk about energy and how it's used to perform work in biological systems. Energy, at its core, is the capacity to do work, whether it's on a molecular level or a physical one. First, there's chemical work. This involves the making and breaking of chemical bonds, which is crucial for building molecules, breaking them down, or even reshaping them in cells. Next up, we have transport work. Imagine this as moving cargo ions, molecules, or larger particles across cell membranes. This is essential for creating concentration gradients, like the difference in sodium and potassium ions across a neuron membrane. Finally, there's mechanical work. This is the physical movement of things, like organelles within a cell, changing the cell's shape, or the beating of structures like flagella and cilia. Oh, and let's not forget muscle contraction. That's mechanical work in action. Energy comes in two forms, and they're both essential for life: kinetic energy and potential energy. Kinetic energy is all about motion. It's the energy of things moving. Think of a rolling ball or blood flowing through veins. Potential energy, on the other hand, is like a battery waiting to be used. It's stored energy. In our bodies, this shows up in concentration gradients, like the buildup of ions on one side of a membrane, and in chemical bonds. Think of glucose storing energy for later use. One amazing thing is that energy can switch forms. For example, the potential energy stored in food molecules can be converted into kinetic energy when you run. However, this transformation isn't always 100% efficient. Some of it is lost as heat. This figure beautifully demonstrates the relationship between kinetic and potential energy using a ball and a ramp. For figure A, kinetic energy is when work is done to push the ball up the ramp. Energy of motion is at play. This kinetic energy is being transferred into potential energy due to the ball's increasing height. For figure B, potential energy is when the ball reaches the top of the ramp and comes to rest. It holds potential energy. This energy is stored because of its position, ready to be released when the ball starts rolling. For figure C, kinetic energy is when the ball rolls back down the ramp. The stored potential energy converts into kinetic energy again. However, the conversion isn't perfect. Some energy is lost as heat due to friction with the ramp and air. This process highlights energy transformations in real-life scenarios, demonstrating how energy is conserved and shifted between forms. This diagram explains how energy flows in biological reactions through exergonic and endergonic processes. Exergonic reactions release energy as bonds are broken. For example, A plus BC plus D plus energy. Some of this energy is released as heat, while the rest is captured by molecules like NADH FOD or NADF as high-energy electrons. Energy storage and transfer capture energy in NADH FOD and NADF is used to form ATP, the cell's energy currency. ATP stores energy in its high-energy phosphate bonds, ready to fuel other processes. Endergonic reactions require an energy input to proceed. The energy stored in ATP is used to power reactions like E plus F plus energy G plus H. This cyclical energy flow highlights how the cell captures energy from exergonic reactions to drive necessary endergonic processes, maintaining cellular function. This slide introduces enzymes, which are essential proteins that act as catalysts in the body. Here's a breakdown. Enzymes increase the speed of chemical reactions by lowering the activation energy required. They make reactions efficient and are vital for maintaining life processes. In reactions involving enzymes, the reactants are referred to as substrates. Enzymes are proteins. Structurally, enzymes are specialized proteins, and their function depends heavily on their specific 3D shape. Isozymes, or isoenzymes, are variations of enzymes that catalyze the same reaction but under different conditions or in specific tissues. This allows the body to adapt the same reaction to various environments or requirements. Enzymes are crucial in every biochemical pathway, from digestion to energy production. This slide highlights a key concept in biochemistry regarding reversible reactions. Reversible reactions. These are reactions that can proceed in both forward and reverse directions, eventually reaching a state of equilibrium. At equilibrium, the rate of the forward reaction equals the rate of the reverse reaction. In the example in the diagram, P plus LPL indicates that two components, P, or protein, and L, or ligand, combine to form a complex. PLP plus L shows that the complex can dissociate back into its individual components. In biological systems, reversible reactions allow the system to adapt and maintain homeostasis depending on the conditions, such as substrate concentrations or environmental changes. Enzymes play a critical role in facilitating these reactions efficiently. This table outlines diagnostically important enzymes, which are measured in blood tests to identify potential health issues. For acid phosphatase, elevated levels are associated with prostate cancer. However, a newer test using prostate-specific antigen has replaced this enzyme for prostate cancer diagnosis. For alkaline phosphatase, high levels may indicate diseases of the bone or liver, often used to assess conditions like bone disorders or bile duct obstructions. Amylase is linked to pancreatic disease, such as pancreatitis, where the pancreas becomes inflamed. Creatine kinase is an increase suggests myocardial infarction, or heart attack, or muscle disease. CK levels help assess muscle damage or stress on the heart. Lactate dehydrogenase is elevated LDH levels, are a marker for tissue damage in the heart, liver, skeletal muscle, or red blood cells, often used in evaluating conditions like heart attacks or anemia. These enzymes provide critical clues to underlying health problems and help guide further testing and treatment. This graph illustrates how pH affects enzyme activity, emphasizing that most enzymes in humans operate optimally near the body's internal pH of 7.4. At a pH of 7.4, enzymes function at their highest rate, efficiently catalyzing reactions. If the pH drifts too far below, acidic, or above, alkaline, this optimal level, enzyme activity decreases sharply. Extreme deviations from the optimal pH can lead to enzyme denaturation, where the enzyme loses its shape and functionality. This highlights the importance of maintaining homeostasis for enzymatic processes and overall metabolic health. This slide categorizes enzymatic reactions into three main types. Phosphorylation involves the addition of a phosphate group to a molecule. It's a critical step in many biological processes, such as activating or deactivating enzymes. Oxidation-reduction reactions, or redox reactions, involve the transfer of electrons. Reduction is where molecule gains electrons, and oxidation is where molecule loses electrons. Hydrolysis dehydration reactions utilize the addition or removal of water. Dehydration reactions is where water is produced as a byproduct when molecules are joined together. Hydrolysis reactions is where water is used to break chemical bonds between molecules. These reactions are central to metabolism and energy transfer in cells. This slide expands on enzymatic reactions, adding two more categories. Deamination is the removal of an amino group from a molecule. Amination is the addition of an amino group to a molecule. Transamination is the transfer of an amino group from one molecule to another. Ligation, or synthesis, reactions involve joining two molecules together, typically using enzymes called synthesis. Examples include fatty acids combine with coenzyme A to form acetyl-CoA, a key molecule in energy metabolism. This slide provides a concise overview of metabolism, which encompasses all the chemical reactions occurring in an organism. Catabolism refers to energy-releasing breakdown processes. Large molecules are broken down into smaller ones, releasing energy. Anabolism are energy-utilizing synthesis processes. Smaller molecules are assembled into larger ones, consuming energy. Kilocalories represents the energy released from or stored in chemical bonds, often used to measure the energy content in food. This slide highlights the essential role of ATP in energy transfer during reactions. High-energy phosphate bond is where ATP stores energy in its phosphate bonds, which is released to power various cellular processes. Aerobic metabolism occurs in the presence of oxygen and can yield 30 to 32 ATP molecules per glucose molecule. It's highly efficient and primarily involves glycolysis, the citric acid cycle, and the electron transport chain. Anaerobic metabolism happens without oxygen and produces only 2 ATP per glucose molecule, primarily through glycolysis. Here are 3 catabolic pathways that produce ATP. Glycolysis is the breakdown of glucose into pyruvate. Citric acid cycle processes pyruvate to generate electron carriers. Electron transport system, or oxidative phosphorylation, uses electron carriers to produce the majority of ATP in aerobic metabolism. In the cytosol, glycolysis starts with glucose breaking down into pyruvate. It produces 2 ATP and generates NADH, which carries high-energy electrons. Other molecules like glycerol and amino acids can also feed into this pathway. In the mitochondria, pyruvate conversion to acetyl-CoA. Pyruvate enters the mitochondrion, gets converted to acetyl-CoA, releasing Co. This step generates NADH for the next stages. In citric acid cycle, or Krebs cycle, acetyl-CoA enters the cycle to produce Co, ATP, and high-energy electron carriers, including NADH and FOD. Fatty acids and amino acids can also feed into this cycle. In electron transport system, NADH and FOD donate their electrons to the electron transport system in the mitochondrial membrane. The electron's energy drives ATP production via oxidative phosphorylation. This process uses oxygen as the final electron acceptor, forming HO. Here, we can calculate overall ATP yield. From one glucose molecule, aerobic metabolism can produce 30 to 32 ATP. This includes ATP from glycolysis, the citric acid cycle, and the electron transport system. This figure illustrates the glycolysis pathway, which is the first stage in glucose metabolism. Here's an easy breakdown of the steps. Glucose activation, steps 1 to 3. Step 1. Glucose is phosphorylated to glucose 6-phosphate. This step uses ATP, meaning the cell spends energy to start glycolysis. Step 2. Glucose 6-phosphate is converted into fructose 6-phosphate. Step 3. Another ATP is used to phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate. Cleavage of fructose 1,6-bisphosphate. Step 4. The 6-carbon molecule is split into 2 3-carbon molecules, dihydroxycetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. DHAP quickly converts to G3P, so now 2 G3P molecules proceed through glycolysis. Energy generation, steps 5 to 9. Step 5. G3P is oxidized, reducing NAD to NADH and attaching a phosphate to form 1,3-bisphosphoglycerate. Step 6. A phosphate group from 1,3-bisphosphoglycerate is transferred to ADP, forming ATP and 3-phosphoglycerate. Steps 7 to 9. More chemical rearrangements lead to the production of pyruvate, generating another ATP molecule in the final step. This figure illustrates the transition from pyruvate to acetyl-CoA and the citric acid cycle, or Krebs cycle, key stages in aerobic respiration. Key steps. Pyruvate to acetyl-CoA. Pyruvate, from glycolysis, is transported into the mitochondria if oxygen is available. Enzyme pyruvate dehydrogenase catalyzes the conversion of pyruvate into acetyl-CoA. During this process, NAD is reduced to NADH. One carbon atom is released as CO. Acetyl-CoA is a 2-carbon molecule that acts as a key input to the citric acid cycle. Citric acid cycle overview. The cycle begins when acetyl-CoA combines with a 4-carbon molecule, oxaloacetate, to form a 6-carbon molecule, citrate. Through a series of enzyme-catalyzed reactions, citrate is gradually broken down, regenerating oxaloacetate. Outputs of the cycle. For each acetyl-CoA that enters the cycle, 2 comolecules are released. High-energy electron carriers are produced. 3 NADH, 1 FOD, 1 ATP, or GTP, is generated. These electron carriers, NADH FOD, will transfer electrons to the electron transport chain, driving ATP synthesis. Electron transport system and its role in producing ATP through oxidative phosphorylation within the inner mitochondrial membrane. Here's a step-by-step breakdown. NADH and FOD, produced during the citric acid cycle and glycolysis, donate high-energy electrons to the electron transport system. These molecules also release hydrogen ions, contributing to the proton gradient. Electrons pass through a series of protein complexes embedded in the inner mitochondrial membrane. As electrons move along the chain, the energy released is used to pump hydrogen ions from the mitochondrial matrix to the inner membrane space, creating an electrochemical gradient or a source of potential energy. At the end of the chain, electrons combine with oxygen and hydrogen ions to form water. This step is critical as oxygen acts as the final electron acceptor, ensuring the continuation of the electron transport system. The accumulation of hydrogen ions in the intermembrane space creates a proton motive force, or a strong concentration and charge gradient. Hydrogen ions flow back into the mitochondrial matrix through ATP synthase, a specialized enzyme. The flow of hydrogen ions through ATP synthase drives the conversion of ADP plus inorganic phosphate into ATP. For every 3 hydrogen ions that pass through ATP synthase, 1 ATP molecule is synthesized. This slide showcases the energy yields from the breakdown of a single glucose molecule, highlighting the differences between anaerobic and aerobic metabolism. On the left, we see anaerobic metabolism, which occurs without oxygen. Here's what happens. Glucose goes through glycolysis, producing 2 ATP as a net gain. The end product, pyruvate, is converted into lactate to regenerate NAD for continued glycolysis. While this pathway is fast, it's not very efficient, producing only 2 ATP for every molecule of glucose. On the right, we explore aerobic metabolism, which requires oxygen. Glucose goes through glycolysis, producing 2 ATP and 2 NADH. Pyruvate is converted into acetyl-CoA, generating NADH and releasing CO. Acetyl-CoA enters the citric acid cycle, producing NADH FOD and more ATP. The high-energy electrons from NADH and FOD enter the electron transport system, where the majority of ATP is generated 26 to 28 ATP. Overall, aerobic metabolism produces 30 to 32 ATP per glucose molecule, making it much more efficient than anaerobic metabolism. In this figure, we see pyruvate at the crossroads of two metabolic pathways, aerobic and anaerobic metabolism. The path it takes depends on the availability of oxygen. When oxygen is limited, pyruvate remains in the cytosol and follows the anaerobic pathway. Here, it is converted into lactate. This process regenerates NAD, a crucial molecule needed for glycolysis to continue, ensuring the cell can produce small but quick bursts of ATP, vital in high-energy demand situations like intense exercise. However, when oxygen is plentiful, pyruvate enters the mitochondria. Inside the mitochondrial matrix, it is transformed into acetyl-CoA. From here, it embarks on the aerobic pathway, entering the citric acid cycle. This cycle generates high-energy molecules, NADH and PHOD, which drive the electron transport system. This pathway produces significantly more ATP, efficiently powering the cell's energy demands. This figure beautifully illustrates how cells adapt to their environment, choosing the best metabolic strategy based on oxygen availability, ensuring survival and function under diverse conditions. This slide highlights the fundamental role of proteins in cellular function. Proteins are the building blocks of life, crafted from an alphabet of 20 unique amino acids. The sequence and number of these amino acids determine the vast diversity of proteins, each with a specific role in the cell. The genetic instructions for protein synthesis are encoded in DNA and RNA. A codon is a group of 3 nucleotide bases that specifies one amino acid in this blueprint. DNA uses the bases cytosine, guanine, adenine, and thymine, while RNA replaces thymine with uracil. This protein-making system, guided by genetic codes, is at the heart of cellular processes, shaping everything from enzyme activity to structural components. This diagram represents the genetic code, as found in the codons of mRNA. Each codon is a sequence of 3 RNA nucleotides combinations of the bases uracil, cytosine, adenine, and guanine that encode a specific amino acid or signal in protein synthesis. The chart is structured to show how codons translate to amino acids. The rows represent the first base of the codon, the columns represent the second base, and the subcolumns list the third base. For example, the codon August not only codes for the amino acid methionine, but also serves as the start signal for translation. You'll also notice certain codons, such as UAA, UAG, and UGA, are labeled as stop codons. These mark the end of the translation process, signaling the ribosome to release the completed protein. This genetic code is universal, serving as the foundation of protein synthesis across nearly all organisms, making it a critical component in understanding biology and molecular genetics. This slide focuses on the process of unlocking DNA's code, which explains how genetic information stored in DNA is used to synthesize proteins. First, we have the gene. This is a specific region of DNA that contains the instructions for building a particular protein. Next, we have transcription, the first step in decoding DNA. In the nucleus, the DNA sequence of a gene is copied into messenger RNA, mRNA. This mRNA serves as a temporary, portable version of the genetic code. Last, we have translation. Once the mRNA exits the nucleus, it heads to a ribosome, the site of protein synthesis. Here, amino acids are assembled into a protein chain in the order dictated by the mRNA sequence. This process involves transfer RNA-tRNA, which brings amino acids to the ribosome. Each tRNA has an anticodon, a sequence complementary to a codon on the mRNA, ensuring the correct amino acids are added to the growing protein chain. DNA directs the synthesis of RNA, emphasizing the key steps involved in the production of mRNA or messenger RNA. DNA strand is used as a template. The process begins with a specific section of DNA being utilized as a blueprint. This template provides the sequence of nucleotide bases needed to create a complementary RNA strand. RNA polymerase enzyme plays a pivotal role. It attaches to the DNA, unwinding its double helix structure to expose the template strand. As RNA polymerase progresses along the template, it pairs complementary bases, guanine with cytosine and adenine with uracil, which is only unique to RNA. This carefully coordinated process forms a strand of RNA, which carries the genetic instructions from DNA to other parts of the cell, where it can guide protein production. Further details and visuals may be presented in the following slides. Imagine a gene, a specific segment of DNA, being activated to produce a functional piece of RNA. This journey begins when RNA polymerase, the key enzyme, attaches to the DNA at a specific starting point. With precision, it identifies the gene's location, binding tightly to ensure accuracy. As transcription progresses, RNA polymerase unwinds the DNA, exposing the section that encodes the necessary information. This creates a template strand, a guide for RNA synthesis. Picture the enzyme sliding along the DNA, unzipping the double helix as it goes, revealing the genetic code that needs to be transcribed. During elongation, free RNA bases, adenine, cytosine, guanine, and uracil align with their complementary DNA bases on the template strand. Unlike DNA, uracil substitutes for thymine here. As each RNA base binds, they link together, forming a growing strand of messenger RNA, mRNA. It's like a precise assembly line where every nucleotide is carefully added to build a chain. The mRNA strand elongates as the RNA polymerase moves forward, ensuring the code is transcribed accurately. As the enzyme travels, the DNA behind it re-zips into its double helix structure, preserving its integrity. Finally, once the RNA polymerase has completed the transcription of the gene, the newly synthesized mRNA detaches from the DNA. This strand of mRNA is now ready to exit the nucleus and head to the cytosol, where it will be translated into a protein. The story of protein synthesis begins where transcription leaves off. Once the DNA code has been transcribed into mRNA, this processed mRNA exits the nucleus and enters the cytoplasm, where the magic of translation happens. Here's how it unfolds. In the cytoplasm, the mRNA strand encounters ribosomal subunits, the cellular machines that will interpret the genetic code. These subunits, small and large, come together to form a fully functional ribosome, creating the stage for protein synthesis. The mRNA, now held within the ribosome, is read in 3 nucleotide sequences called codons. Each codon corresponds to a specific amino acid, and this is where transfer RNA, tRNA steps in. tRNA molecules, carrying individual amino acids, have anticodons that match the mRNA codons. Think of tRNA as couriers, delivering the right amino acid based on the genetic instructions. As the ribosome moves along the mRNA, codon by codon, tRNAs bind to the matching sequences. Each tRNA deposits its amino acid, and the ribosome links these amino acids together to form a growing peptide chain. The process continues in a cycle, with the ribosome shifting to the next codon and welcoming a new tRNA. The tRNAs, after delivering their cargo, leave the ribosome to be recharged with another amino acid. Meanwhile, the peptide chain elongates, forming the backbone of the final protein. Eventually, the ribosome reaches a stop codon on the mRNA. This codon signals the ribosome to release the completed peptide chain. The ribosome subunits disassemble, and the mRNA and tRNA are recycled for future use. The newly synthesized peptide chain may undergo folding and modifications to achieve its functional form. At this point, the gene's instructions, once locked away in DNA, have now become a fully formed protein, ready to perform its role in the cell. We examined how living organisms harness energy. Plants capture radiant energy from the sun, while animals rely on consuming energy-rich molecules like carbohydrates and fats. Chemical reactions are the backbone of life processes, involving the making and breaking of bonds to convert substrates into products. Enzymes act as catalysts, speeding up chemical reactions essential for life. They lower activation energy and are specific to the reactions they facilitate. The interconnected web of chemical reactions in the body comprises metabolism. We covered catabolism, or breaking down molecules to release energy, and anabolism, or building molecules using energy. Finally, we delved into the central dogma of molecular biology. DNA provides the blueprint, transcribed into mRNA in the nucleus. This mRNA is then translated in the cytoplasm, using ribosomes and tRNA to assemble amino acids into proteins, the workhorses of the cell.